In the first few stages of cellular respiration, our body converts all of our key nutrients into our reduced electron carriers of NADH and FADH2. The electron transport chain then is our key stage in our third stage of cellular respiration, where we can take those electrons from our NADH and FADH2, transfer them to ultimately oxygen, and in so doing, we're going to create this electrochemical gradient of protons that will allow us to produce ATP. So ultimately, all of our catabolic pathways, whether we're breaking down sugars or proteins or fats, will all lead into this electron transport chain that will allow us to use those oxidation reduction reactions to produce energy. The electron transport chain will involve 4 different key protein complexes, as well as many different smaller redox cofactors that will be involved in the transport of electrons through each of these different protein complexes. So we'll take a look at each of these different complexes and see what their role is in creating that electrochemical gradient of protons that we'll then use to produce ATP. So we'll start with complex 1, which is also called the NADH ubiquinone oxidoreductase. So as the name suggests, our complex 1 is going to transfer electrons from NADH to our ubiquinone, or coenzyme Q. So ultimately, we're going from our reduced NADH and our oxidized coenzyme Q, or ubiquinone, to our reduced coenzyme Q, or ubiquinol, and our oxidized NAD+. Our complex 1 will has 2 different key components. These are formed from multiple different protein subunits. So you can notice that the slightly different colors here represent the individual protein subunits that make up this larger protein complex. We have this membrane arm that consists of a series of transmembrane proteins, as well as our matrix arms that sticks out into the aqueous environment of the mitochondrial matrix. So we have our intermembrane space marked as the P side out here, and then our protein stretching out into the matrix side. Again, the matrix side is where kind of all of our reactions occur with our citric acid cycle, as well as fatty acid oxidation. That's then producing that NADH and FADH2 that will be feeding into our electron transport chain. So our pathway here starts with NADH dumping the 2 electrons into complex 1. These electrons are going to get excited FMN, or flavin mononucleotide. Again, FMN is a single or double electron carrier. It has that similar structure to FADH2, just lacking that single adenine nucleotide. So it's a mononucleotide as opposed to the dinucleotide of our FADH2. They then transfer those electrons through a series of iron-sulfur centers. So again, we've got our, you can note the structure of our FMN here, as it's using this head group to transfer those electrons. And then we'll be passing those through each of these iron-sulfur clusters, ultimately to our coenzyme Q. So we transfer through those iron-sulfur clusters. We showed the structure of those previously. We use the iron as our electron acceptor and donor. So we're changing the redox state of that iron, ultimately dumping our electrons onto our lipid-soluble coenzyme Q. So that's, of course, the redox portion of our reaction. Now, again, the main goal of our reaction here is to produce an electrochemical gradient. So we want to produce that electrochemical gradient where we have high proton concentration on the outside in the intermembrane space and a low proton concentration on the inside in our mitochondrial matrix. So while we are doing this redox chemistry here, we want to use the energy generated from this series of redox reactions. So this will be a spontaneous reaction overall. We're going from lower reduction potential here at our NADH. So NADH has the lowest reduction potential of the redox cofactors here, that is, to our higher reduction potential of our coenzyme Q. Again, remember that a transfer from lower to higher reduction potential is always spontaneous. So that spontaneous reaction is going to be used to help us push protons out of the mitochondrial matrix and into our inner membrane space. This is going to be accomplished through our membrane arm. So we use a conformational change. So the membrane arm shifts and triggers the matrix arm shifts and triggers the membrane arm, which will then force pump 4 protons from the matrix side. So we would represent that as 4H plus N. So these are protons on the matrix side, the N side that are getting pumped out. So we're losing 4 protons from the matrix side and adding 4 protons on the intermembrane space side. So again, that will create a larger concentration of protons on the outside and start to create that electrochemical gradient. At the same time, we're also going to take protons from the matrix side and add them to our ubiquinone as we reduce it to ubiquinol. So our body is really clever about grabbing those protons off of the matrix side so that we continue to add to the electrochemical gradient even as we are reducing and forming our ubiquinol here. So the overall reaction here, we are transferring 2 electrons from NADH to ubiquinone. That's our coenzyme Q. And at the same time, we are transferring protons from the matrix to the intermembrane space through that triggered conformational change. We have 4 protons transferred for each NADH that reacts. So our overall reaction then is NADH plus ubiquinone and 5 protons from the matrix side reacting to produce NAD plus, our reduced ubiquinol, and 4 protons on the intermembrane space side. So you can note the balance of our equation overall. We have 5 positive charges on the right, 5 on the left, and of course we have 6 protons overall. We can't be consuming or producing protons, of course, because we have to conserve mass. We're just shifting the balance of protons on the inside in the matrix space versus proteins on the outside in the intermembrane space. Now, so far, we've only managed to collect electrons from NADH and feed them into the electron transport chain through our ubiquinol or coenzyme Q. But of course, our fatty acid beta oxidation and our citric acid cycle also produce reduced electron carriers in the form of FADH2. So we need a way to take electrons from FADH2 and transfer them into the electron transport chain. And so what we'll do is we'll use our ubiquinone again as our electron carrier. So this is going to be our shared gathering point of electrons from both complex 1 and complex 2. So we can say both complex 1 and complex 2 will be taking their electrons and feeding them into the formation of our ubiquinol, or QH2. And then our remaining complexes after that will take that ubiquinol and continue to transfer the electrons from there, ultimately to oxygen. So our complex 2 is succinate dehydrogenase, and our goal is going to be to transfer electrons from FADH2 to our ubiquinone to produce that ubiquinol. Now, of course, the name here should sound familiar because this is the enzyme from the citric acid cycle. So this is succinate dehydrogenase. It's what converts succinate to fumarate in our citric acid cycle. And it's actually bound to the membrane of our mitochondrial matrix. So we have, again, a transmembrane portion right here shown embedded in the membrane, as well as our portion on the mitochondrial matrix side. Our succinate will come in here and react and immediately form that FADH2 that we will then use to transfer electrons into the electron transport chain. So remember, right, FADH2 is a prosthetic group. Which means, of course, that it's bound tightly inside of proteins and it doesn't freely move around by itself. So in order to transfer these electrons from our succinate dehydrogenase to the electron transport chain, it actually has to be a part of the electron transport chain, essentially. So we can't just take that FADH2 out of our succinate dehydrogenase and bring it over to our complex 2, like we did with complex 1. Instead, we make the succinate dehydrogenase itself part of our electron transport chain. So from there, we're going to proceed in a very similar manner to complex 1. So we transfer those electrons from FAD after it's produced from succinate. We go through, again, a series of iron-sulfur centers. These transfer electrons one at a time down to our ubiquinone, our coenzyme Q, producing our ubiquinol. Now, notice we also have one final electron carrier here. So that's our heme B from our cytochrome B. So we showed that structure previously when we looked at our different redox cofactors. And the role of our cytochrome B here isn't 100% known, but it's thought to act as maybe a electron carrier to help catch electrons that might try to slip out from our ubiquinone. So one of the issues that we'll talk about in more depth later is that once we produce these sorts of reduced electron carriers like our ubiquinol or even our NADH, they can potentially give those electrons away to oxidize other molecules, which can then cause some of the oxidative damage. So this cytochrome B here of our heme group isn't directly involved in the electron transport chain, but it just helps to catch extra electrons that might otherwise escape and cause oxidative damage. Now, ultimately, as you'll notice here with complex 2, we are, of course, doing our usual redox chemistry and transferring electrons from our electron carrier to the coenzyme Q, but there's no movement of protons here, right? You don't see a long membrane arm that's pumping protons out. You don't even see protons getting added to our ubiquinone to create that ubiquinol, because, of course, those protons are actually coming from our FADH2, which is pulling those protons off of our succinate, right? So we're not consuming any protons in our mitochondrial matrix. We're just taking those protons as hydrogens off of our succinate. So complex 2, unlike complex 1, doesn't actually contribute to our electrochemical gradient, which is directly why our FADH2 doesn't produce as much energy as NADH. So complex 2 doesn't add to the electrochemical gradient, resulting in less energy per FADH2 than NADH. So, of course, we've used that general rule of 2.5 ATP per NADH versus 1.5 ATP per FADH2. And this difference here between complex 1 and complex 2 is directly what causes that difference in ATP production. Okay, so at this point in our pathway, right, we had both complex 1 and complex 2 producing our reduced ubiquinol. So now, of course, complex 3 is going to take that ubiquinol and we'll transfer that onto ultimately complex 4. So we're going to transfer it to cytochrome C, which will then transfer it to complex 4. So both the electrons from complex 1 and complex 2 will feed together into complex 3, which will then feed into complex 4. So here's the overall structure of complex 3. So this is also called the ubiquinone to cytochrome C oxidoreductase. You can notice the similarity in naming for our complex 1, where it's an oxidoreductase because it's transferring electrons from ultimately our ubiquinone, coenzyme Q, to cytochrome C. And you can see in the figure here, right, we have multiple different protein subunits. We also have many different electron carriers from our porphyrin ring heme groups to our iron sulfur centers that will all act to transfer those electrons through to our cytochrome C, which will also contain, of course, that porphyrin ring heme group, but is formed in or is contained inside of a soluble protein that will then carry those electrons to complex 4. Now, the transfer of electrons inside of complex 3 is a little bit more complicated compared to our previous pathways, and it follows what we call the Q cycle. So experimentally, what we see is that 4 protons are going to be transported across the membrane per every 2 electrons that reach cytochrome C. And you might remember that cytochrome C is a single electron carrier, so we're actually going to be transferring 2 electrons to 2 different cytochrome C units. So 2 of those 4 protons that we transfer are going to come from our ubiquinol. The other will ultimately come from our mitochondrial matrix. Our Q cycle is a good model to explain how those 2 additional protons end up getting picked up. So the end result of that will have 2 molecules of QH2 becoming oxidized, releasing protons into the intermembrane space, and then 1 molecule becomes re-reduced, thus resulting in only 4 protons being transferred per reduced coenzyme Q. Okay, so to understand this Q cycle better, we're going to walk through each stage of our Q cycle. So here's stage 1. So in our complex 3, we've got this main chamber here marked in green, where we're going to be accepting electrons and transferring electrons between our ubiquinone, our coenzyme Q. Then we're going to send those electrons out to cytochrome C to ultimately transfer those to complex 4. So initially, we're going to bring in our reduced ubiquinol, QH2, and this will come in to transfer electrons to cytochrome C. Now, the issue that we have here is that cytochrome C only accepts 1 electron. Ubiquinol can, of course, transfer either 1 or 2 electrons. So what it's going to do is transfer 1 electron over to cytochrome C. So that's 1 of our 2 electrons. But the other electron is going to go through these heme B groups over to an oxidized ubiquinone group. So our oxidized ubiquinone accepts 1 electron and becomes our coenzyme Q radical with the single electron transfer, while of course at the same time this QH2 is then fully oxidized to ubiquinone, and we pump those 2 protons out into the intermembrane space. So we have essentially 2 different binding sites here for our coenzyme Q. One where we bind the reduced coenzyme Q, our ubiquinol, one where we bind the oxidized coenzyme Q, our ubiquinone, and so we're able to release the protons only out onto the intermembrane space side. So the overall reaction of our first step of the Q cycle is 1 reduced coenzyme Q is consumed, along with 1 oxidized coenzyme Q, and of course our oxidized cytochrome C. We produce our reduced cytochrome C, an oxidized coenzyme Q, and our partially oxidized coenzyme Q, as well as releasing those 2 protons from our ubiquinol into the intermembrane space. As we head into the second step of our Q cycle, so now we already have our partially oxidized coenzyme Q, and we're going to bring in a new unit of our reduced ubiquinol, or reduced coenzyme Q. So we bring this in and we do our single electron transfer as before, right? 1 electron going out to another unit of cytochrome C, 1 electron going in towards the mitochondrial matrix through those heme B electron carriers. So this electron will go to our partially oxidized coenzyme Q, or partially oxidized, partially reduced, to now fully reduce it to ubiquinol. So we're going to take 2 protons from the matrix side to add to that partially reduced, partially oxidized coenzyme Q. So we're actually consuming protons from the matrix side, right? That's consumption of 2 protons from our matrix side, since we have this matrix binding site of our coenzyme Q, while at the same time we're also releasing 2 protons, again from our ubiquinol into the intermembrane space side. So our overall reaction, we're now consuming that reactive intermediate of our partially reduced coenzyme Q. We're consuming a second fully reduced ubiquinol. We're also grabbing 2 protons from the matrix side and oxidizing another, or taking another oxidized cytochrome C and reducing it to our reduced form. But we're producing a QH2 and a fully oxidized coenzyme Q, as well as, of course, releasing those 2 protons onto the intermembrane space side. So overall, when we put these both together, right, we brought in, again, 2 units of our reduced ubiquinol, but we only ended up consuming 1 overall, right, because we produced a new unit of ubiquinol, as well as 2 units of ubiquinone, but we also consumed a unit of ubiquinone. So the net consumption is just 1 ubiquinol to produce 1 ubiquinone, while, of course, oxidizing or taking 2 of our oxidized cytochrome C and converting those to 2 reduced cytochrome Cs, again, since they each carry 1 electron, taking 2 protons from the inside while releasing 4 protons on the outside. And notice in our proposed Q cycle mechanism, we're actually not pumping any protons through. We're just releasing them using this kind of clever mechanism where we always bind our coenzyme Q in the reduced form, our ubiquinol, on the intermembrane space side, and we're always binding that more oxidized form of our coenzyme Q, our ubiquinone, on the matrix side. So the matrix reactions happen up there, which then means that we consume the protons from the matrix side, and the oxidation reactions happen at the intermembrane space side to allow us to then continue releasing those protons outside. Right, so there's essentially a net transfer of, there's a net gain for our electrochemical gradient, that is, of 6 protons total, since we're losing 2 on the matrix side and producing 4 on the intermembrane space side. So at this point now, our electrons are all in our cytochrome C, right? From whether it started from NADH or FADH2, they both dumped into ubiquinol, which is now dumping those electrons into cytochrome C. Cytochrome C is a little bit unique in our transport chain. So it's the second mobile electron carrier. So our ubiquinone was the first electron carrier, but ubiquinone, since it has that lipid nature, only moves through the membrane. Whereas our cytochrome C is actually an aqueous soluble protein that will move through the intermembrane space. So it's now going to be able to move through the fluid of our intermembrane space over to the next protein of our complex. So it's a soluble heme-containing protein that has this heme group with our iron, of course, in the center that can accept or donate that single electron as it changes our redox states. So again, as usual, we're switching back and forth between our ferrous or ferric, iron 3 or iron 2 states as we transfer that electron. And our goal then is to carry a single electron from our complex 3 to our cytochrome oxidase, which is complex 4. So complex 4 is the final step in our electron transport chain, although you can also technically consider complex 5 or the ATP synthase as an additional component of the electron transport chain. But complex 5, of course, is really doing the oxidative phosphorylation, where we're taking the electrochemical gradient and using that to produce ATP. So complex 4 is the last point where we're transferring electrons in order to create that electrochemical gradient. Now, as you look at the structure of our complex 4, so we are going to be taking those electrons, of course, from our cytochrome C, and we're going to be oxidizing cytochrome C and taking those electrons and transferring them to oxygen to produce water. We're going to be transferring those electrons again through heme groups with our iron in there as our electron carrier. But we're also going to be using some copper groups coordinated with our cysteines and our sulfur as well. Our complex 4 forms multiple units that are combined into a dimer. So you can notice if I cut this right down the middle, we have these two halves that look essentially the same. So these are the two monomers of our dimer, but each of these monomers are made up of many smaller subunits as well. So let's take a closer look at what's going on inside of one of the monomers of our dimer. So this would be duplicated in the other half of our dimer. So we have our cytochrome C is going to be binding over here on the intermembrane space side. Again, this is a soluble protein that will move through the intermembrane space, through that aqueous environment, and bind on the intermembrane space side. We'll then take those electrons and transfer them in. So we go through those copper atoms that will accept those electrons, transfer them to our heme groups. So this is a iron copper center. We have the copper transferring to our heme, and we have another copper involved here as well. And this will ultimately catalyze the reaction with our oxygen to then produce water. The production of that water will require 2 protons from our matrix side. So these protons will flow in and add to our oxygen to create that water molecule. As we produce that water molecule, we're also going to be pumping protons from the matrix to our intermembrane space. We have some protons getting consumed and some protons getting pumped out. Now, notice this is happening for a single water molecule. So for making one water molecule, that would require 2 electrons coming from cytochrome C. In order to oxidize a full oxygen molecule, so 1 O2, that would require 4 electrons or 4 units of cytochrome C. So it's 2 electrons per water molecule or 4 electrons per oxygen molecule, since, of course, half of a diatomic oxygen molecule is all we need to make our water. So in terms of our overall ratio here, then, it is 1 ubiquinol for 2 cytochrome C for 1 water molecule. Or, of course, we could say 2 QH2 to 4 cytochrome C to 1 oxygen molecule. Again, of course, because there's 2 water molecules for every 1 oxygen molecule. So at this point, we've completed our transport chain. So again, we had NADH or FADH2 feeding into our ubiquinol. Our ubiquinol feeds into cytochrome C, which then feeds into the production of water. So we've now completed our overall cycle. We've looked at each of the 3 different, or each of the 4 complexes, NADH and FADH2 inside our complex 2 as our key starting electron donors, leading to coenzyme Q, leading to cytochrome C, and then finally ending at oxygen getting oxidized to water. In terms of our overall pathway, so from complex 1 to complex 4 versus we of course have to consider complex 2 to complex 4 separately, since we have basically 2 branches of the pathway that come together and then go through complex 3 and complex 4. In complex 1, we consume 1 NADH, we consume 11 protons from the matrix side, and we consume 1 oxygen molecule. We produce 1 NAD+, 10 protons on the intermembrane space side, and 1 water molecule. From complex 2 to complex 4, we had our FADH2 reacting with 6 protons from the matrix side and one half of an oxygen molecule, that is, one oxygen atom, to produce our oxidized FAD, 6 proteins on the intermembrane space side, and 1 water molecule. Now, to break down the numbers of these protons a little bit more here. So again, of course, complex 1 took a total of 5 protons from the matrix side when we count the protons from our from the transfer of protons across, as well as the protons that went into our ubiquinol. We then had complex 3 as our next step in this pathway. So complex 3 takes 2 protons from the matrix side as it produces our ubiquinol. So that's, of course, from the reduced ubiquinol that we produced, since we have 2 that are consumed and then 1 that's produced ultimately. And then complex 4, which ends up consuming 2 more protons, or pumping 2 protons across, as well as consuming 2 protons that are used to make our water molecule. As far as the protons that are produced on the intermembrane space side, so complex 1, of course, pumped those 4 protons out. Complex 3 released 4 more. So again, this was from the 2 units of ubiquinol that each released their protons onto the intermembrane space side. And then complex 4 had 2 more protons that were pumped out as we reacted our oxygen and produced water. For complex 2 to complex 4, of course, these numbers are just the same numbers from complex 3 and complex 4, the 6 and then the 6 that we had above, because complex 2, of course, doesn't produce or consume any protons on the matrix or the intermembrane space side. This 6 to 10 difference in terms of the number of protons that are produced on the intermembrane space side is directly resulting in the difference in the number of ATP units synthesized for FADH2 and NADH. So at this point, it looks like we have a complete picture of the transfer of electrons from NADH and FADH2 to water as we create that electrochemical gradient of protons. However, if you're paying close attention, you might notice a couple flaws here, right? So our NADH could only come in from the mitochondrial matrix side, right? So this is only mitochondrial NADH. On the other hand, of course, our FADH2, since this is a prosthetic group, right, we only really took FADH2 from succinate dehydrogenase. And of course, the citric acid cycle that would be generating that FADH2. Our fatty acid beta oxidation and any other pathways that produce FADH2 wouldn't be feeding those electrons into our succinate dehydrogenase. So how do we take cytosolic NADH or FADH2 from other pathways and feed it into our electron transport chain? So for FADH2 from other sources, we're going to feed it in through a couple of different pathways. And these will use other proteins, so essentially other components of our electron transport chain to feed electrons into ubiquinol. So we're still going to use ubiquinol as our universal kind of electron dump. So everything from NADH or FADH2 is always going into that ubiquinol. But we're just going to add additional proteins that will transfer those in there. So on the inside in our mitochondrial matrix for our fatty acid beta oxidation, so fatty acid beta oxidation will produce FADH2 through our acyl-CoA dehydrogenase as we convert our fatty acyl-CoA, that's our alkane form, to our enoyl-CoA, that's our alkene form. Now, this is a soluble protein. It's not part of the, it's not a membrane-bound protein, but it will take that FADH and it will transfer those electrons through an electron transport flavoprotein that will also have a FAD prosthetic group. And this will then transfer to a ETF-Q oxidoreductase, again, using our usual naming convention there. The starting electron donor, ETF, our electron transport flavoprotein, and our ubiquinone, our coenzyme Q, and this will ultimately transfer those electrons to our ubiquinone. So this is essentially a complex 2.5, if you will. Although nobody uses that name, so that's just, you can think of it as essentially another form of complex 2 dumping those electrons into ubiquinol. On the cytosolic side, we also have a couple of other sources that will be dumping electrons into ubiquinol. So we have this dihydroorotate dehydrogenase that has a prosthetic flavin mononucleotide that will dump electrons into QH2. This comes from some of the biosynthetic pathways for nucleotides. And then we also have this glyceryl-3-phosphate dehydrogenase that contains a prosthetic FAD that will dump electrons into QH2. Glyceryl-3-phosphate dehydrogenase, of course, you might recognize. It does the reaction from glyceryl-3-phosphate to our dihydroxyacetone phosphate that we use when we're breaking down glycerol. Now, I do want to take a little bit of a closer look at that glyceryl-3-phosphate dehydrogenase because it can also play a role in shuttling NADH into the electron transport chain. So again, for our NADH, we have a pathway for mitochondrial NADH, but not for cytosolic NADH. So if we want to transfer cytosolic NADH through, we can use a couple different shuttle pathways. One of those is the glyceryl-3-phosphate shuttle. So our glycolysis will produce NADH. That, of course, is from our GAPDH, or glyceraldehyde 3-phosphate dehydrogenase. That can then go through our cytosolic glyceryl-3-phosphate dehydrogenase, which can use that NADH to reduce our dihydroxyacetone phosphate to glyceryl-3-phosphate. So at that point, we are kind of running this pathway backwards from what we usually see, but this allows us to produce the NAD+ so that we can then continue glycolysis, while this glyceryl-3-phosphate can then go to our mitochondrial glyceryl-3-phosphate dehydrogenase, which is bound on the outside of our inner mitochondrial membrane, and this will then convert it back to dihydroxyacetone phosphate, while producing FADH2 that can then feed into our ubiquinol, which will then continue our pathway with complex 3. So again, we have another essentially complex 2, if you will, where we're feeding from FADH2 into our ubiquinol, although in a sense, this is actually a variant on complex 1, because we are taking NADH now and feeding it into ubiquinol, but in this case, we're feeding it from the cytosolic side instead of from the mitochondrial matrix side. Now, of course, this also results in the transfer of those electrons from NADH to ubiquinol without pumping any protons out. So this glyceryl-3-phosphate shuttle reduces the energy production from NADH, right? Because ultimately, if we're not creating as much of an electrochemical gradient, we're not going to be producing as much ATP. Essentially, at this point, we're going to be reducing the energy production of NADH to the same level as FADH2, right? Again, FADH2 normally enters the pathway without pumping any protons out, so we only get the protons that are pumped out through complex 3 and complex 4. Now, of course, the other mechanism of shuttling NADH into the mitochondria we've already looked at before. That's through this malate aspartate shuttle that uses our malate alpha-ketoglutarate transporter and our glutamate aspartate transporter to transport that aspartate out that we need for the urea cycle, as well as transport malate back in that we need for the citric acid cycle. So when we combine these together, we get our malate aspartate shuttle, where we effectively consume NADH on the cytosolic or intermembrane space side and produce NADH on the mitochondrial matrix side, leading to essentially a transport of NADH into the mitochondria. And of course, we've looked at this shuttle before when we looked at the urea cycle, so I won't talk about each of these reactions in detail, but it is another mechanism for moving that cytosolic NADH into the mitochondria, allowing us to then go through complex 1 and get our full energy production from NADH, unlike that glycerol 3-phosphate shuttle. Ultimately, this is the reason why we say that glucose produces 30 to 32 ATP, because it actually will depend then on whether you use the glycerol 3-phosphate shuttle versus the malate aspartate shuttle. All right, so at that point, we've, of course, fed all of our electrons into producing water and producing this electrochemical gradient of our protons. So again, we have our chemical gradient in terms of the difference in proton concentration or pH across our membrane. We have our electrical gradient in terms of the high positive charge outside and high negative charge inside. Now we can use that gradient in complex 5 to generate ATP. So that will be the focus of our next lecture.